Hello everybody, my name is Frank. I'm from Leipzig University, as you see here. And we are going to share, let's say, roughly an hour or so, where we talk about active particles which are controlled by feedback interactions. And I'm trying to show you how you can do that. I show you some examples as well of how to apply that, and I try to give you a bigger background on why to do that maybe. And that is the first question we would like to answer. So why the hell would you try to feedback control active particles? So first of all, we just have to negotiate somehow or come to a common ground what I mean with feedback systems. And therefore, let me show you this kind of simple slide. So on the left side, you see a sketch of how I imagine a feedback system. It actually consists out of two systems which are coupled here together. And one of the systems is obtaining some input. It is processed and maybe also stored in any kind of way, and it's typically causing some output. It could be kind of anything. This is very abstract at the moment. But if you take now that output signal and process that as well in this kind of system F here, and you might introduce some latency there, some delay, then you can couple that back to the input and either add that to the input or subtract that from the input. So if you have that additive part, then you would call that a positive feedback. If you have that negative part here, you would call that a negative feedback mechanism. And by doing that, you have this kind of circuitry here where these two systems are coupled to each other and are strongly influencing each other. And most of the times it's not really clear whom is influencing whom. They are both strongly coupled together, right? So accordingly, negative and positive feedback have different kind of outcomes. So there are all kinds of phenomenologies connected to that. So on the negative side, so if you have negative feedback, that typically leads to adaptation because you subtract from the input signal something until the output signal is not any more different from the input signal probably, and that means you can regulate something onto a specific level. So this is often done with negative feedback loops. Positive feedback loop leads in general to an amplification. So you add something in addition to that, the signal gets bigger than the original signal. So the output is also getting bigger and so on and so on. And this can yield exponential growth, for example, at the end, and instabilities. But if you mix both of them, you can have certain stages which are realized, certain set points very easily. You can have polarization effects, oscillation effects, all kinds of different scenarios are happening if you mix positive and negative feedback. And there are a lot of examples where you actually have these positive and negative feedback loops realized. So if you think of technology nowadays, there's in every cell phone so many feedback cycles which control your energy consumption, which control the brightness of your screen, or in a car, the cruise control is a typical example. But you also have lots of feedback systems in nature, and the climate is, for example, a typical feedback system where you have on the water circuit, so water which is actually flowing somewhere, evaporating, going into clouds, and the clouds rain down as water again at different stages or at different positions, and that are causing a lot of climatic effects at the end. So since we are interested in active particles, we would probably shift a bit more to the biological side, because our active particles mimic somehow biological activity. And therefore, let's have a look at examples which are a bit more on the biological side here. The first one comes from actually the ecological side. So this is an ecological feedback example, which you certainly have heard of, which describes the relation between predators and prey, right? So imagine that you have a certain population of prey and a certain population of predators, and these populations are measured by these variables x and y. Then they interact, of course, in a certain way. Typically, it's that the predator eats the prey, and therefore the predators can grow in population and the prey decreases in population, and that is described here by these Lotka-Volterra equations, which are actually nothing else than a feedback cycle between these two populations, or two coupled differential equations here. Yeah? So this is the change in the population of the prey with time, and this is the change of the population of the predator in time. And the change in the prey population has something to do with the prey growth rate, which is alpha here, and then the probability to get eaten by the predator, which is captured here by beta, and the y is just the amount of predator that's available currently. And in the same way, the amount of predator, or the population of the predator, is determined by the natural death rate here, gamma, of the predators. But there's also some growth rate, and the growth rate is captured here by that sigma, because at that moment the predator has eaten some of the prey. Of course, it can replicate and then increase the population. So if you solve these coupled differential equations, you get this kind of oscillating behavior, which you've seen. So first of all, the prey population is increasing because you have that growth rate here. At a certain stage, the predator sees, oh yeah, it's nice to have you, and eats all the prey. And then, of course, the amount of prey is decreasing again, and the predator is rising. But if there's not enough food anymore, also the predator will die. So this is a very simple ecological model, but it's also capturing a number of true observations here. For example, for that snowshoe rabbit and the lynx, which are shown here in this nice little picture. And here you see the population over the years, and you see this clear oscillating behavior and even this tiny little phase shift from time to time. But of course, this kind of experiment is pretty more difficult. Yeah. So at the end, everything behaves like this kind of limit cycle oscillator, which runs around a certain fixed point, which is the average population of predator and prey you have. Yeah. So this is a simple ecological feedback example, and a simple physiological one is, of course, what I described also in the lecture notes, which you've probably learned about in your school lectures, is the glucose level in your blood, which is maintained by feedback cycles at a certain level, which is around 90 milligrams to 100 on 100 milliliters here, somewhere around that. So if you start to eat something, of course, your glucose levels will rise, and the pancreas is then releasing insulin actually to store the glucose in your liver or in any kind of body cells for later usage, because you want to have the blood sugar level kept constant. But at a time when you just have a lack of glucose in the blood, then the pancreas is releasing another hormone, which is glucagon, and the liver releases the excess glucose, which is stored actually to the blood, and trying to keep that concentration level constant. So all of these regulating processes lead to something which is called in biology homeostasis. So keeping up a certain set point of all of your processes in the body, like body temperature. There's a homeostasis for that. Is done by feedback loops actually, and this is not only happening at the level of your body or a whole organism that can go down into the individual cells, and already in the cells it's quite complicated. So here is that a part of the signaling chains in a cell. Of course, when you have on the left side certain signals that are interacting with proteins and enzymes in the cell, and they cause certain processes to happen here. For example, one of the important ones is the family of RAS proteins, which are belonging to the family of G proteins that are multifunctional proteins, which control, for example, cell differentiation, cell growth, and also cell death in a way. So once they are activated by some process, they can cause cell growth. And in many patients that have cancer, you find an increased level of activated RAS proteins. So they need to be activated by some kind of cofactor. And in many cases, you have an increased level of these mutated RAS proteins that are active all the time. And therefore, you probably have a cancerous situation there in these patients. So this is another example, and I can't tell you about all of those details because actually I'm not a biologist, but you may easily think of that as a kind of a feedback cycle, a very complicated feedback cycle, or a computational device, or whatever you would like to imagine that. So at the end, if you want to conclude somehow on that, for a moment at least, is that you can describe all these living systems as a collection of feedback cycles. But in these feedback cycles, there's one big important issue playing a role. This is activity. And as you probably or most likely learn through that training here, activity is really important because you put in energy all the time, and that keeps some of the processes running and easily steerable. And that's one of the important things. So summarizing at that moment, your body or your cell is a collection of very complicated feedback cycles that scientists, of course, try to decipher. And beyond that, there's probably something we can do also with active particles. So a whole organism can be probably also simplified down to a level where you consider this kind of beautiful fish into a sensing device here. This is your input, a processing device, which is your brain or your neural network. There's some kind of activity. So this could be our active particle at the end. Here the active particle is a living fish, of course, and I have a hard time to influence that fish information processing, or even to influence the activity of the fish in an experiment. This is a difficult thing to do, but I can probably more easily do that with a synthetic particle. And that's one of the reasons. Another reason is that there's some interesting things beyond just these complicated feedback loops. And that is something you've seen probably many times during the individual lectures here. This is the collective behavior. So if you have your body, this is not made out of a single cell. If you have even a single cell, that is not made out of a single molecule. It's made out of many, many different types of molecules. And they are playing like an orchestra in a nice way, synchronized in a way that everything works out. Yeah? So at the lowest level, you still have Brownian motion, noise, and everything present. And on the macroscopic level, everything arises as very nicely deterministic. And there's an important thing behind that, and that is the signaling, of course. Cells signal and do synchronize in a certain way, differentiate in a certain way, and they do that not, of course, based on physical and chemical interactions, but the signaling is introducing something more, that a cell which is at a certain distance can still react to that other cell somewhere here without having a direct contact. Yeah? So that becomes more prominent if you go to larger length scales, for example, these flocks of birds or these schools of fish. The fish, of course, do not interact because they have necessarily any kind of physical contact, even so they may feel the other one's hydrodynamic flows. They exchange information. So these informations that you exchange by signals, I call them beyond physical interactions or information-related interactions, because there's not really a direct physical force between them. And they are really a great invention because they release every human being or every fish or every biological species from the laws of physics. So still I feel the gravity every day in the morning, but I'm still not running against the wall or the closed doors because I see them. And if I at least open my eyes, and that's some additional interaction I have. But the door is not influenced by that kind of seeing in any kind of way. So the door stays force free while I move to a new position. Yeah? So you can easily induce changes in these systems just by information. You can tell the fish to go away if I bring a shark very closely, and they would certainly try to escape, and without any kind of additional force you have to put in. Yeah? So this is some important difference as compared to many other physical systems where you want to create a change, you need to have a lot of energy actually to do that. Here the fish are doing that for you. Yeah. So these new beyond physical interactions, information-based interactions are very, very interesting as they play apparently an important role in biological systems. And from that comes the inspiration actually for the answer, why would you like to control feedback, active particles by feedback? So I give you 3 examples here to understand collective phenomena based on information based interactions, to explore the mapping of those interactions to any kind of physical descriptions you people have developed or the science have. So you know, for many systems there's a statistical mechanics description which you can use for large ensembles. And the question is if any of these also applies there, and can we understand that based on physical laws which have been formulated. And the big question there is the closeness of many biological systems to critical phenomena where you are at a point where you're especially responsive and how this comes about. So this is one of the emerging questions there. And of course, at some point, you probably also want to integrate these feedback loops into synthetic active matter. So far, we are doing that only, or to a large extent only, with the means of computer control. But you see how beautifully nature has evolved in a way, biological species that can do things where everything is orchestrated from the smallest objects to the largest length scales. And this is completely in contradiction to what you currently do with material science. Material science, you take a big piece of silicon and saw that into thin sheets, and in these thin sheets you groove or structure transistors and wires and whatever to create a computer, while your brain is perfectly organized in a completely different way. So you see that kind of contradiction, and probably the feedback control of active particles and what you get out of that can help to understand or to go new ways in this direction. Okay. So what's the plan? The plan is now that I talk a bit about that synthetic replacement of that fish here on one side and what I need for active particle feedback control. So on one side, I will need a switchable self-thermophoretic swimmer, and most likely Ivo will taught you about autophoretic swimmers already quite a lot, so I don't have to do quite a lot on that. But I will still tell you how our swimmers work, which we can control by feedback. Then we'll tell you about the feedback control and how you characterize that and what's the accuracy and what are the limitations, and also some experimental details there. And I give you some applications which come from more the physical side, which is how to measure active particle pressure, some very fundamental thing. Then what you can do in the self-organization of active particles by feedback-controlled interactions. And since I have a lot of time, actually, I also highlight a bit how you can incorporate techniques of machine learning into the behavior of swimmers at the end. Okay, so this is the program. And of course, there is no program or lecture without help. And the help comes here from 3 sides if you want to. You don't have to. So on one side, I have prepared a tiny little Python notebook, which you can run in the Jupyter environment. Of course, you have to get familiar with that if you want to do that, but you can still have a look at that. It's linked also in the lecture notes, so you can directly click and execute it in your browser, and it deals with photo nudging, active particles, and so on. If you're not really familiar with Python and also the Jupyter notebook environment or whatever, which is related to that, you can actually also look at the Python course I gave for bachelor students essentially. So there are lots of examples also, including even machine learning, but mostly it's physics related. So you will have to have a look at that. On the right side is the exercise I thought you might be interested in, so the kind of experiment you shall have a look at, especially at the experimental results. You will analyze the motion of active feedback oscillators. This is done here in a Pluto notebook. I will give you the presentation of that. You will get the data. You will have to do something with that. So that is the kind of plan we have. And so why not start with topic number 1 and show you how these swimmers work.